Hi everyone, I'm Pam Allenstein, Plant Collections Network Manager at the Association, and I will be your staff representative during this virtual conference session. Before we begin, here are a couple of tips to get the most out of your virtual conference experience. First, if you are experiencing poor video or audio quality, we suggest closing other programs on your computer, especially other browser tabs. Your inbox will still be there at the end of the session, I promise. If you have any other problems or questions, association staff are available to help. Give us a call at 610-708-3010 or send us an email at info@publicgardens.org. Second, while this session has been pre-recorded, the presenters are here with us today to answer questions, and we'll be happy to do so in a live Q&A portion after the pre-recorded session. At some point during the session, the presenters may activate a poll. Poll responses are anonymous, so please be sure to let us know how you really feel. Finally, you can submit your questions using Zoom's Q&A function that you will find at the bottom of your Zoom taskbar. Questions can be submitted anonymously, and then you can also upvote other questions to indicate that you would like to hear them answered. We will try to answer as many questions as we can during the live Q&A portion of this session. Some questions may be answered directly through the Q&A feature. Thank you and enjoy today's session. Hi everyone, I'm Pam Allenstein, the Plant Collections Network Manager at the American Public Gardens Association. Over the years, my work's been involving promoting excellence in plant collections management and also a continent-wide approach to conservation. And I've noticed especially in recent years, that there are sometimes common tree species, at least according to what might be available conservation rankings, that they are secure in the wild, that become threatened or even endangered in a relatively short time span. And now we've seen that globalization is really ramping up introductions of deadly pathogens and pests. Some of them are really wiping out trees in the wild and in our gardens. And while we are planning for disasters like hurricanes, floods, fires, planning for a disaster that is caused by a biological invasion is really different, and it's going to require different kinds of techniques to address them and to mitigate the impacts. They really present a continuous assault, requiring us really to adapt strategies, even over decades, to manage the impacts on our trees and in the wild and in our gardens. And as we've all learned in recent months with dealing with the COVID-19 pandemic, which is another form of biological invasion, acting early can really significantly flatten the invasion curve and can mitigate the impacts on our forests and our gardens. So we know that public gardens, working together with a broad community of government agencies and other stakeholders, researchers, can really make a difference in addressing these biological invasions while also contributing to conservation efforts. So this session is going to feature 4 different case studies. They're from different regions and focused on conserving a tree species that's threatened by a pest or pathogen causing high rates of mortality. So some of these threats have been with us for a long time, for decades, while others are relatively new on the scene. So you'll hear about conservation activities at different stages. The first case study will be featuring Richard Sneetzko. He is a forest geneticist at the Dorena Genetic Resource Center in Oregon. That's part of the U.S. Forest Service. In his research, he's focused on developing durable disease and insect-resistant populations in forest trees, and he frequently partners with public gardens. So Richard will be sharing his work with conserving Pinus albicollis, whitebark pine, and developing a durable resistance to rust pathogen. Our second case study is going to be from Shauna Walsh. She's the conservation biologist at the National Tropical Botanical Garden. She leads the garden's efforts in implementing Hawaii strategy for plant conservation and conducts applied research in plant breeding systems, pollination ecology, and population genetics. And Shauna's case study will focus on her work in collecting, banking, and reciprocating seed of Kauai's native Metrosideros species that are threatened by a fungi causing rapid ohia death. And our third case study is going to be given by Kevin Conrad. He's the curator of the Woody Landscape landscape plant germplasm repository at the U.S. National Arboretum. I've had the privilege of working with Kevin for a number of years in his role as the representative from the USDA's Agricultural Research Service in collaborating together to build our plant collections network program. In recent years, we've been looking for opportunities to work together to build new collections, and this is one of our newest initiatives. Kevin will be describing it, and it's focused on preserving the germplasm from our native sassafras, which is threatened by a disease called laurel wilt. Laurel wilt is actually a serious disease which is threatening all of the laurel family, Lauraceae genera. It has already killed large populations of red bay, swamp bay, and avocado in the southeast. If it moves to the western part of the country, it could also impact Oregon myrtle and California bay laurel, as well as the avocado industry in California. And finally, we have a fourth case study, which will be given by Tim Thibault. He's the curator of woody collections at the Huntington Botanical Gardens in San Marino, California. Tim's initiated many field collecting expeditions, including projects that have targeted southwestern oak species, Arizona walnut, and a number of magnolia species. He's part of a working group organized by the U.S. Forest Service targeting western species of Fraxinus that are threatened by emerald ash borer. Tim will be focusing on a project to collect fragrant ash from Arizona and describe plans for future work in collecting additional southwestern Fraxinus species. So with that, I will be turning it over to the case study presenters. Thank you.
Thank you for the opportunity to present at this virtual conference. I hope to share some important information with you and some of the partnerships that we have going on. What I'm going to talk about today is whitebark pine, a high elevation species. I'll talk about more in a minute about that. But specifically what I'm going to talk about is using genetic resistance to white pine blister rust for restoration work. And a key part of that is partnerships, including with groups like yourself and whatever like that too, because very few of these programs have succeeded worldwide, and there's some keys to success, and you'll see partnerships are a big key. Okay, whitebark pine is a conifer species of high elevation western North America, the US and Canada. The map on the right shows the range of the species. And then you can see some photos of the species from Southern Oregon and different things. But it can be it's a foundation species, it's a keystone species, very, very important species of these high elevation environments. It is under threat, however, by climate change, mountain pine beetle, and white pine blister rust. And white pine blister rust is the part I'm going to talk about today and how we solve that part of the equation. Okay, as we all know, invasive pests or diseases are all over the place now, and we're having to confront them all the time. And many things are tried along the way, trying to exclude them, trying to slow the spread. But for many of these, it will eventually come down as do our native species have genetic resistance? Can we harness that genetic resistance? How do we do that effectively and quickly and get it back out on the landscape? And that's pretty much what I'm going to talk about. The U.S. Forest Service got involved in white pine blister rust about over 50 years ago. And developing for western white pine and sugar pine and eastern white pine, looking at those three species and in terms of whether they could find and deploy resistance. Your organization has produced signs like this to help educate the public, and that is a huge part of the program, to let the public know what the problems are with our forest out there, why the problems exist, and whether there's actually solutions. Because, you know, it's one thing to say there's a problem if you can't say there's a solution. So information like this and sharing it with the public can be vital in terms of supporting these programs. A program like mine, you will see, is kind of narrowly focused in some regards, and we don't have the access to the many thousands or hundreds of thousands of people that your group does in terms of conveying this information, sharing this information, garnering feedback from the public and public support. Okay, so the problem I'm going to talk about is white pine blister rust. This pathogen came in over 100 years ago and has decimated a lot of our populations of white pine species. There's 9 species of white pine in the US, 4 of those occur in Canada. 8 of those species occur in the West, including the white bark pine. This species, you might say, how does it even exist? Because it needs 2 hosts. It also needs a ribes or currant or gooseberry host. There's a few other genera involved as well too. But the disease spreads from the ribes to the pine and from the pine back to the ribes. So it's an intricate system. In the center photo there with the red and green, you see some trees alive and you see some trees dead. And that's really the end game for resistance. Each row there is a plot of 10 trees of seed from a different parent tree. We now have screened 1,300 parent trees. And you can see I told our statistician at work, I said, hey, Doug, I don't need your help on this. The green trees are the good ones, with the resistance, the red ones not very good for resistance. So there's some dramatic resistance that exists in our own species. We're talking about natural resistance here, not genetic modification or anything like that. I'm not putting a value judgment on that, but at least in the species, the first look is to natural resistance. Even with genetic resistance, it takes something to put this together. There's a lot of research programs around the world that have looked into resistance, and there's been nothing come of it because they haven't been linked with the key players. One of the key players is, my job is to build applied populations of resistance and get it out there to the field. So I fall into kind of that tree improvement or tree breeding realm. On top of that, there's a reforestation and restoration, all the end users. Without them, it doesn't matter what I develop. There have to be people interested in using it and people willing to find a way to get it back out on the landscape. In addition, we need management support because none of these things happen overnight or without some funding. So it's only where these 4 components come together that you have success. The programs we've developed at Drina Genetic Resource Center with the Forest Service are easily in the top 5 in the world because they've integrated these components, including the partnerships. Okay, Drina Genetic Resource Center is located near Cottage Grove, Oregon, has facilities for doing this type of work. It was set up to do this type of work. We're involved in some other things as well too. But in the bottom left-hand corner, you'll see what our inoculation chamber is. It's a fog chamber. We can produce a fine mist fog where you can put your hand out and not see your hand. It could be the world's largest fog chamber for this type of work. And that makes it very easy to do things at an operational schedule as opposed to a research schedule. So we're interested in finding populations of trees, not single cultivars and put back out in the forest. Again, I mentioned there's 9 species of white pines in the US. All of them, to a certain extent, are threatened by this disease. They're all susceptible to this disease. The key is to develop populations, not a single cultivar, and get it back out there. To do that, we need to unite with various other entities, such as yourself, and conservation education and keeping support for these programs is vital. Okay, where is the resistance? Is it everywhere? Is it at the same level everywhere? And the map on the right-hand side of the slide shows Oregon and Washington, which is our main part of the focus of the range, although we work with some others as well. And in there, you have these pie charts. And the key thing about the pie chart is there's different colors from green, blue, orange, pink, and red. And the summation of these is the pie charts that have a lot of green and blue. That means a lot of the parent trees we have tested from those areas are resistant. The ones that have mostly red, orange, and pink, such as the Olympic National Forest, there we found very little resistance so far. So even when you find resistance, it's not necessarily going to be everywhere. And the key is you may say, so what? Why don't you just move stuff from Southern Oregon up to Canada? Well, in forestry, you've also got to take into account adaptability of the species, durability of the resistance, and also maintain a genetic variability as well too. So in many cases, we won't move white bark pine from a distant source up to another source without good reason to do so. So we've really subdivided the range into different seed zones and trying to find resistance in each of those zones. Okay, partnerships. I mentioned that's real key. This is a list of a number of the partners, and you can see there's universities, there's a government organization, there's nonprofits, tribes, et cetera. They all help. One example, American Forest a couple of years ago, we were going to have the largest cone crop in about 10 years in white bark pine. Many years you don't get a cone crop. So even if you're going to do resistant work, you need seed, you need cones. And we were going to get this great cone crop in 2018, but we really didn't have the funding to do the collections. And American Forest stepped up and helped provide some of the funding. And with that, we were able to capture this once in a 5 or once in 10 year cone crop. In these slides, you can see some people climbing trees, some people bagging the cones on trees, some people planting trees. In many of these cases, these aren't Forest Service people. These are people from different organizations. Okay, a case study is kind of Crater Lake National Park. If we had had this conference in Portland, as we were originally going to, some of you would have had a chance maybe to visit Crater Lake National Park. And Crater Lake National Park has whitebark pine, and the map on the left shows a range of whitebark pine in Crater Lake. And then the little squares, the little dots, show the trees that they've collected cones off of, sent to Dorena. We've grown the seedlings for 2 years. We've infected or inoculated them with the rust. We've scored them for up to 5 years after that. We've rated them for resistance. And then the real key step is this middle slide shows a restoration planting using the knowledge from this testing. It was planted about 10 years ago. If any of you have been to Crater Lake, there's a place called Rim Village. That's where just about everybody parks. So nearly all 400,000 visitors to Crater Lake each year will see this planting. So it's a restoration planting, but it's also a genetic planting because every one of those seedlings has a tag that tells what parent tree it came from. But we'll know whether these parent trees are still able to give us seedlings that are suitable under a changing climate as well as resistant to the white pine blister rust. Okay. And then where does your group come in? A few years ago, we had some extra seedlings of whitebark pine and some of the other species we were working with. And we had outreach back and forth with the public gardens, and we shipped some seedlings of whitebark pine and some other species to the gardens that they could use for conservation, education, and research purposes. So as I said, you guys have a much larger outreach to the public than we do. And so this will be vital for us, as well as maybe seeing how the whitebark does in these different environments as well. Okay, now how can we speed all of this up? And I don't know if some of you are science fiction fans and follow Star Trek. I did from the early days. And one device that always fascinated me on Star Trek was the tricorder. And basically, a tricorder is a handheld device by which you can tell different things about different environments or different people or different trees, et cetera, too. So what we're trying to develop with Ohio State University, this is an early phase, but can we, instead of waiting for cones to be produced on the trees, collect a couple of needles out in the wild, survey thousands of trees, tell which ones very quickly are resistant, and then build resistant populations and get it back out for restoration. So this is really great. Eventually there'll be more funding for this, but right now we have a tech development proposal to do the early phases, but that's the dream. Develop a thing where any resistance, any insect or disease that come up with any other species, how fast can we turn this around and find the natural resistance that's out there? Okay, the other thing we want to do with whitebark pine, and there's some people have been clamoring to do this, is to sequence the genome. The genome of conifers is like 8 times the size of a human genome. Huge. So very few of these genomes have been sequenced, and only in relatively recent years. But now people want to sequence for whitebark pine. What's holding it up now? Basically funding. There are people jumping at to do it. There are people capable of doing it, networking to doing it. So we're just waiting funding. So certainly if you know some sources, I can put you in contact with players. Once the genome is sequenced, we can develop genomic resources to aid the development of disease and insect resistance, as well as to aid restoration guidance under a changing climate. So it's a powerful tool that will leverage what we already know with traditional breeding, silviculture, ecology. Okay, so really when we have an invader, a new disease, a new insect come in, we can envision the future. We can do nothing, for society, or we can do something. And in this case is where there's invaders, genetics really is the key. There's a lot of people that talk about some other elements, but almost none of those elements, biocontrol, other management things, almost none of those will work unless you have an element of genetic resistance. So rather than waiting to the end, the key is to look into genetic resistance early on. I show here a slide of the Whitebark Pine Foundation, links at the top, both in the US and Canada. You can go there for more information on whitebark pine. And then I list a series of other references about resistance to white pine blister rust, also about the microbiome of whitebark pine and some of the genetic variation factors in whitebark pine as well too. So there's a lot of data being gathered. This is a species that has been proposed for listing under the Endangered Species Act. And of course, we can acknowledge all sorts of people, all the groups that are partners and cooperators, certainly the Forest Service for supporting our efforts, and then some of my colleagues across different organizations have provided photos. So I hope I've given you the story of whitebark pine, I think it's an early success. You know, it still needs to play out. We still need to get it back out in the landscape. That's happening now. But I think it could be a real success story, but it will take partnerships to do that. Thank you very much.
Aloha, everyone. Thank you for tuning in virtually. And Pam, thank you so much for inviting me to be a part of this session. I should preface by saying that there are many, many people across Hawaii, the US, and the world that are involved in work to mitigate against and combat rapid ohia death. So I'll just be sharing some of the work that the National Tropical Botanical Garden has done to safeguard ohia on Kauai threatened by rod. And just a bit of background on ohia. That's the Hawaiian name for all native species in the genus Metrosideros, and they occur from sea level to high elevation bog habitats. And the genus in Hawaii is comprised of 13 taxa, representing 5 different species. And they are the most abundant and ecologically important tree in Hawaii. As a foundation species, they provide the habitat upon which all other native plants and animals depend. They form the largest portion of the canopy in our native mesic and wet forests. And so I mentioned they provide the habitat for a lot of our native animals, and that includes our native forest birds and snails. And as you see in these photos, are all pictured in ohia. It is also one of the first trees to colonize fresh lava substrate and is therefore instrumental in the process of soil development and ecological succession. And it's very culturally important in Hawaii as well. It comes up in many mo'olelo or stories, mele or songs, oli, which are chants, and is one of the kinolau or physical manifestations for many of our Hawaiian deities. The first time I entered an old growth ohia forest, I think I was a teenager or in my early 20s, but I was mesmerized by their stateliness. And I had never seen or heard so many native forest birds in the misty canopy, all the different types darting from one blossom to another to feed on nectar remains very clear in my memory. And that was on Hawaii Island. And on Hawaii Island in 2014, large numbers of ohia were observed dying off. And it was something different. And it turns out this death was being caused by what was later realized to be 2 different species of fungal pathogens, previously unknown to science. And in 2018, they were described as Ceratocystis huliohia, and that specific epithet in Hawaiian translates to changes the natural state of ohia, and Ceratocystis leucoohia, which translates to destroyer of ohia. And the disease caused by these pathogens was named rapid ohia death, or ROD. And once infected, leaves turn yellowish and brown within days to weeks and die. So you have forests that look like this. And as you can imagine, just by looking at this photo, very negatively impacting all other native plants and animals that depend on ohia forest. And since it was first detected on Hawaii Island, it has destroyed over 175,000 acres of ohia forest on Hawaii Island, or also known as the Big Island. And at the end of 2016, a statewide ROD strategic response plan was published. And part of that plan includes collecting and storing seeds of all 13 Metrosideros taxa and safeguarding them in seed banks as a genetic safety net. And so members of the Hawaii Seed Bank Partnership worked together to develop a seed collecting protocol for ohia. And ohia seeds are orthodox, so they can be stored long-term in dry, frozen conditions. And part of that work that the partnership did was to develop seed zones for each of the main Hawaiian islands on which ohia occur. These were not previously established in the islands. And seed zones are areas within which plant materials can be transferred with higher likelihood of survival. So they're a crucial tool to guide collections and reintroductions. And NTBG staff, several of us worked together with our state botanist, our Kauai Island state botanist of the Department of Land and Natural Resources, to delineate the zones for Kauai based on climate, vegetation, and our local knowledge, boots on the ground knowledge of the island. And in mid-2017, it wasn't yet detected on Kauai, but anticipating that it was only a matter of time, NTBG sought funding to collect, bank, and reciprocate seeds of the 4 native Metrosideros on Kauai. And 2 of those taxa are endemic to Kauai. And when I said reciprocate, that's with other facilities, seed bank facilities, as a backup. And this was all in case of the future potential likely spread and potential need to reforest areas to come to the disease. And we were awarded a grant from the Hawaii Tourism Authority, actually, at the end of 2017 to do this work. And the grant also supported some community outreach and education about ROD, but I won't have time to get into that today. But it was only several months later, in May 2018, that Ceratocystis huliohea was detected on Kauai. And then later that year in December of 2018, the other fungal pathogen, leukohea, was found. And this map is showing the distribution of detections on Kauai to date. And so our work on that grant to collect seeds of all the Metrosideros on Kauai from all the seed zones began in early 2018 and continued through mid-2019. And many of our work trips were helicopter access, so we could reach the interior of the island and all those different seed zones. And some others were drive and hike access. This was a photo of my colleague Natalia and I. It was a helicopter access overnight fieldwork trip collecting ohia along a ridge on the island. And my colleagues Tim Flynn and Dustin Wolkis, this is along a drive and hike access trail in Kokee. And my colleague Ken Wood and I here got flown into a remote area of the Waimea Canyon region, also known as the Grand Canyon of the Pacific, and camped collecting ohia. And then here, my colleague Merlin Edmonds and I were on the summit of a place called Namolokama. I had never been to this place. It's only accessible by helicopter, and it's often shrouded in clouds, so it's really difficult to get to. And we camped there collecting ohia and almost didn't make it out because of such poor visibility for the pilot. But during all of our collecting work, we were making bulk collections and also collections from individual plants, which we tagged in the field with a unique ID, and that ID system standardized across the state. And specifically, that's to allow us to link individuals found to have ROD resistance back to the mother plant from which those seeds were collected. And what we're looking for in the field for fruit ripeness is these dried capsules that are cracking open with these very tiny golden seeds. And turns out it's actually quite challenging to collect ohia seeds. One of the reasons being in the taller statured forests, you need a pole picker. You guys, a lot of you I'm sure are familiar with climbing or using pole pickers. But the other thing is that when these seeds are wind dispersed, so we also have to time it when they open and the seeds are there, as you see, and there's not too much wind. So it's kind of a team effort. One person's using binoculars to look in the canopy for this, and guiding the person with the pole picker, which hangs on to the cut branch, and that person gently lowers it down to get those seeds. And then once seeds are brought back to the seed lab and associated data is entered into our database, they're removed from capsules. And my colleague, seed bank manager Dustin Wolkis, rubberized this food processor as a way to extract seeds from capsules very easily. And to date, from that work, we collected nearly 9 million ohia seeds. And I was doing some measurements of how, to give a better visual of how many seeds what that means. So basically, if all of those seeds germinated, that equates to roughly 20,000 acres of what a ohia forest, give or take, is structured as. And that's actually about half the size of Washington, D.C. So not huge amount, more work remains to be done. But again, this is just work that NTBG did to collect and bank ohia seeds. There are others across the state doing this work. And the two photos on the right are just to give a visual. These little dishes are about that big. And so that's what about 1.2 million seeds looks like. So pretty tiny. And then 1/3 of the seeds we're banking on site in our seed banking lab. And then the other third we're reciprocating with our colleagues at the Department of Land and Natural Resources. Their seed banks on another part of Kauai. And then 1/3 on the island of Oahu at Lion Seed Conservation Lab. And this table is showing our project targets for that grant and our actuals with a percentage of goal met. So we were able to deliver on most of those targets, but fell short on being able to get collections from as many individual trees as we set as a goal. Another cool thing we were able to do is provide hundreds of thousands of seeds from dozens of accessions to researchers investigating rot resistance, and so for our 2 Kauai endemic Metrosideros. And that research is ongoing. And just wanted to wrap up by sharing some available resources. The Rapid Ohia Death website, hosted by the College of Tropical Agriculture and Human Resources at UH Manoa, includes a lot of info and education and outreach materials, such as brochures, pamphlets, recommended practices, activity books, including, I saw something Pam shared with me, the APGA Plant Heroes Rod Field Guide and Rod Activity Book. So I was happy to see that up there. And this was really a great resource. I really liked it, and learned that Plant Heroes is part of APGA's plant protection program, and that's made possible with support from the U.S. Forest Service. So I encourage you to check that out. And as you're hearing in this session, biological invasions are threatening many important trees all over the world, and I encourage you to get informed about possible pathogens in your area so you can help limit the spread and support work being done to safeguard your forests. So mahalo, thank you for your attention. And if we don't have time for questions, feel free to email me. Aloha.
Thanks, Pam, for introducing me and offering this wonderful opportunity to talk about this terrible problem, biological invasion. And sadly enough, we're talking about sassafras today. You would think that sassafras is a ubiquitous species in the United States, and it wouldn't be under a threat by a biological invasion, but unfortunately that's not the case. But I got ahead of myself. Let me go back one and acknowledge Pam Allenstein. Pam's work with me on this project has been instrumental in its success, and I'll cover it a little bit further as we go through the process. So what about sassafras? Again, ubiquitous across the eastern United States. I consider it a workhorse in ecosystems, both for wildlife and for soil stabilization and development. So it's really a great tree. It's also a great ornamental. Beautiful color. When you plant it in the landscape, plant it young, and it'll do really well for you. And you can cook with it. It makes great filet. So why is it a problem? Well, then there's laurel wilt. So in 2002, laurel wilt was discovered in Georgia. And since then, it's killed over 300 million red bay trees, principally in Florida. So its impact is tremendous. As you can see, it's 90% mortality rate in many species. As far as sassafras is concerned, the story is still unfolding as far as its impact, but it does have an impact. It does affect the genetic diversity of this species. So when you're thinking in terms of plant conservation, ideally you want to affect change before the loss happens. So with laurel wilt, it's another introduced problem. The red bay ambrosia, excuse me, red bay ambrosia beetle is introduced from China, and you can see some of the other impact. The lower left-hand corner, the death that you'll see of red bay throughout the range of the invasion. But really the problem and the challenge is in this slide. So when you look at sassafras and you look at the distribution range, it is across the eastern United States. And then the map on the upper right-hand corner shows the impact from 2002 to 2015. So again, it's pretty significant in that time frame. And then the lower right-hand corner, you see the 2019 map. So you can start to see how it's starting to jump. You can also see comparisons between the distribution of the disease and the infestation and the distribution of the host plants, red bay and sassafras. Look at Arkansas as an example, Kentucky and Tennessee, and you get to start to see the problem that we have with conserving sassafras. And the way that Pam and I formulated the response and the efforts at conservation is collaboration. And that's one of the underlying themes of today's talk, or talks. And with sassafras, it was essential. When you think in terms of the distribution range of this species, Trying to do it on your own is next to impossible, if not impossible. Having spent the last 16 years trying to working in germplasm conservation, conserving a plant of this nature with this kind of extensive distribution range is quite the challenge. So collaboration. And let me go back for a moment. Collaboration in this case starts with the American Public Garden Association and the National Arboretum, and a part of the National Arboretum is the Agricultural Research Service. And I'll go on I'll go on in a little bit on them a little bit. So the American Public Garden Association, you can see a correlation there, excuse me, between the range of sassafras and, of course, the distribution of public gardens in the association. So it lends itself to collaboration. Think in terms of trying to conserve a species when the process involves multiple visits to any one population. And then, of course, as Pam mentioned, the Plant Collections Network, which is a core program within the American Public Garden Association focused on germplasm conservation and working with professionals within that network who understand the need, the process, and ultimately the goals of this effort. And then there's the National Plant Germplasm System. We're part of the Agriculture Research Service. The National Arboretum is a part of the National Plant Germplasm System. And its main focus, excuse me, the main focus has been or is the conservation of the conservation of genetic resources for American agriculture. In our case, plant genetic resources. So the National Arboretum, what do we bring to the table? Years of germplasm conservation and the efforts involved in it, but also plant geneticists who are focused on understanding the genetic diversity of species they work with. So in this case, sassafras, the first part was collection. The second part is understanding what we're trying to conserve. And then there's the National Center for Genetic Resource Preservation in Fort Collins, Colorado. That's a part of the National Plant Germplasm System and focused on long-term conservation in storage. So sassafras being suborthodox, meaning it will store for several years without losing significant viability, but after that it starts to drop dramatically. So that offers challenges to us as far as conservation. How do we conserve it? It would be wonderful if it were orthodox, we could just put it in the freezer and be done with it. But in this case, we have to go through an extra effort. Fort Collins' effort, for the National Center for Genetic Resource Preservation's effort is to figure out how we can store it, either through tweaking seed storage, making it possible, or if necessary, using cryopreservation techniques. So the sampling process. That's really where this collaboration takes an effort that could take careers, meaning decades to accomplish, to where we have made significant inroads in a very short period of time. So the way we've approached this is that we put the call out for collaborators, for cooperators within the American Public Garden Association. The idea was to find natural populations occurring what I refer to in the backyard of the public garden. If they can go out their back door to a naturally occurring site of sassafras, they can collect not only leaf samples at any time they want, but also seed or budwood if necessary. And then the issue of ownership, finding the owner of the land and getting the necessary permissions is eliminated. We have that in store without any effort. So again, using the public garden community, we've been able to streamline the process significantly. I would add, so 23 cooperators, 16 states, but I would want to mention we could use additional samples needed from Ohio, Virginia, South Carolina, Illinois, Arkansas, so on and so forth. So if you're in, or if you have sassafras growing naturally in your public garden and you'd like to be a part of this process, we'd be very interested in talking with you. And here again, here's the players at this moment, 23, the process of collecting seed, they send it to us, and then we work on using collaboration again with the National Arboretum and the researchers there, we'll figure out what the genetic diversity of the species is. And once we do that, then we'll be able to go back and determine collecting protocols, where we want to focus our efforts, where we want to collect. We assume, and it's a significant assumption, that given the range of the species, that it will have a fairly diverse genetic map. But we really don't know. And in order to do it right, we have to figure that out. So once that's done, then we'll be able to write the collection protocols. Finally, once that's done, the ultimate goal is to put it in storage. And that again is where Fort Collins comes in. Their long-term seed storage effort and cryopreservation effort lends itself to this type of work. And again, it's critical. So you think in terms of so many species that have been impacted by a biological invasion. Chestnut, the American chestnut, American ash, is, to name but a couple. So if we can affect change now and conserve the genetic diversity of these species before the impact is so significant, then ideally or theoretically, we would be able to repatriate the genetic material back to its area of origin, which is unheard of. You think of in terms of chestnut blight and the American chestnut, if we had the science and if we had the technology going back to when that was sweeping the country, what effect we could have changed. So again, I'd just like to thank Pam for all her help in this process. Thank everyone for contributing. Of course, the American Public Garden community for all of its efforts. If there's anything I can do, you've got my email address, and I'd be glad to help in any way I can. Thank you.
All right, thank you, Pam, for inviting me to be a part of this panel. To my fellow panelists, it's a real honor to be sharing the virtual stage with you. And to the audience, thank you. I know you have choices in your work, and I'm glad you feel that this is important and are choosing to spend your time in this way. I've been asked to speak about a case study involving southwestern ash species. Let's be honest here, it's a little surreal at this point. Just got the grant end of March, and due to various reasons was not able to do the field work scheduled for this year yet. So this is going to be a little different. I'm going to talk about the concept, and I'm going to encourage you to get off the sidelines and get into the game. The original brainchild came from Richard, who we heard from first. He founded the Western Fraxinus Working Group, realizing that our species were susceptible and there's a pest coming. My concept in this grant was to marry two ideas. Being able to get to a plant species while it's still common and has good genetic diversity, but we know that that's a limited time offer. So my conservation mentor pictured here was Michael Wall. He was curator of the seed bank when I was at Rancho Santa Ana Botanical Garden. And he was the one who really preached the gospel of common to me when everybody else was talking about looking for rare things. Let me tell you, I brought in an accession that was under 30 maternal lines and 2,000 seeds. I had to explain myself to Mike Wall. Other authors have contributed more recently. Sean Hoban worked with Fraxinus excelsior in the UK, and he found that 30 seeds from 30 maternal lines was a good genetic hit on the population. Personal conversation with Sean, he's also stressed getting multiple populations and really focusing on maternal lines more than the number of seeds per. But both Sean and Mike are saying 30 maternal lines. When we think about the nature of Fraxinus, most of the species are dioecious. So if you want to get 30 maternal lines, if we're going to assume a 1-to-1 sex ratio, we're looking at least 60 trees from a population to be able to meet our genetic target. And that's a fairly common species. And here it is pictured. It's coming for us, and our western species are susceptible. Now, I'm sure you've all seen this map or one like it. From an initial outbreak in the East Coast, we see a general radial spread. But what I want to point out is that big jump that happened across the Midwest to Colorado a few years back. Now, if it does this jump again, it's going to be in Arizona and New Mexico, and that's my wheelhouse. So again, when Richard is proposing the Western Fraxinus Working Group, I'm thinking, what can I do? I maintain the permits to collect on public lands throughout Arizona and New Mexico. This is where I can contribute. And if we look at that jump again, anecdotally, in the early days of infection of emerald ash borer, there were localized outbreaks around NASCAR tracks. Reportedly, people bringing their own firewood for tailgating and then leaving it. I'm not going to say that that's going to be how this is going to get to Arizona, but let's just take it as an example. The Arizona Raceway is highlighted with the green arrow. And if we take the expansion from the recent epidemiology paper by Ward et al., we see that we go from Phoenix into those populations in the mountains surrounding very, very quickly. So the final piece of the puzzle was a series of conversations with Jeff Karstens. He's curator of the Fraxinus Germplasm Repository at USDA Ames. He really argued for a small, manageable project. So we kicked around a few ideas, and Fraxinus cuspidata has about 7 populations on public lands that I can get permits for, about 6 on Forest Service lands, and 1 in BLM. So again, it's like eating a box of chocolates. They're all good. It doesn't really matter which one you start with. So why not start with the pretty one? My degree's in ornamental horticulture. And let's do the flowering ash first. But again, this is hopefully part of a longer term process. So that's the concept. And this is how you can get involved. The low-hanging fruit is the APGA U.S. Forest Service Tree Gene Conservation grants. Couple of things to note. Ashes would not apply here because they're orthodox seed. This is looking for things that can't be readily seed banked. The other thing that I would draw your attention to is you need a minimum of 3 distribution sites to safeguard that material that you've collected. And it says ideally 6 or more. You want as many as possible. If you do this work and you do it well, it's about like growing zucchini in your garden. You hope you have a lot of neighbors you can give out a lot of extra zucchinis to. So I'm going to assume you've got the plant side relatively well in order. You have your desiderata list. You can say, I want to collect this. And if you're going to be going for looking for things that are common but experiencing threats, there's a bunch of sources of information. Okay, so this is the same tab on the APGA website where you find the Plant Collections Network. Next one down is the Plant Protection Network. So they've got a series of pages on insect pests of concern in North America. And one of the things I really appreciate is if you're more comfortable in Spanish than English, they can help. Local regulatory authorities are also fine sources of information. Los Angeles County did up this cute most wanted poster for insect pests that we do not want here in LA County. And you can see that emerald ash borer is already on there. And as you're doing the permitting process, you're going to be working with agencies that produce large reports. This is 100+ pages here. And they're, you may be able to find something that they're concerned about and help them. So don't be afraid to look at those resources and talk with those folks as well. So I'm going to conclude here. This is a set of plant groups that would easily meet the APGA U.S. Forest Service grant criteria. And you can see that Quercus is kind of the poster child of what we would be hoping for. It's got a multi-site collection, and there have been, Pam, I'm hoping that I counted correctly, 13 grants to date on for oak collecting through this project. Walnuts, just above it, I'm I've had the opportunity to participate in as well. I'm glad to see there's 3 total grants there, but no PCN collection. Maples, very charismatic, already has its PCN collection in place. Asian longhorn beetle, coming straight for it. To my great surprise, no APGA U.S. Forest Service grants to date. And I do have an asterisk on this one because we just heard from Kevin. Lauraceae does not have a PCN collection, in spite of being charismatic, but no official grants from this fund. And I'm also aware of Chad Washburn's work with the genus Persea, but looks like we could use some more proposals in that area as well. So with that, I'm going to wrap it up and throw it back to Pam, and I think questions next.
Hi, everyone.
Hello, everyone. I'm Richard Duran, your association's Marketing and Communications Manager. Over a decade ago, the association launched an awareness campaign with Rainbird called National Public Gardens Day, celebrated the Friday before Mother's Day to focus attention on the important role of
Hey, everyone. Sorry for that technical glitch. I think we'll be coming back to Richard Duran a little later on. But we wanted to open up the Q&A portion of our session. First of all, we were hoping to find out a little bit more about you. If you could answer this poll question, does your garden have an active seed collecting program? We'd really like to know whether you are already involved with seed collecting. And just mentioning, if you could use Zoom's Q&A box to post your questions, that would be great. And while we're waiting for folks to post questions for the presenters as well as answer this poll, I'd like to ask Richard a question. How successful would you say these genetic resistance programs are, and what would you think would make them more successful? You spoke about keys to success.
Hi, Pam. Thank you for the question. You know, there are some programs throughout the world, New Zealand, the US, Canada, that are very successful. One of the key points is to keep the programs applied and not to have them get caught up in just research. The others pull in, you know, cooperators. And I always make a point that there's been 14,000 selections made in Porterford Cedar for testing. And other than one population at Redwood National Park, I didn't make any of them, but I'm responsible for testing them. So we have a lot of partners that we pull in. And the slide I showed with the Venn diagram with the 4 components: research, applied tree improvement, you know, the restoration managers and the administrators and managers are all key. They must all come together, and public support is a big part of this. So the biggest thing is not to have it get caught up in research, or find somebody who's done this successfully. I'm talking with the people over in Hawaii. So there you go.
Fantastic, fantastic. Thank you so much. And thanks so much to folks who responded to the poll. It looks like we've got the results, and I'm sharing the results with you now, that over 63% of you are indicating that you've got an active seed collecting program. And that's fantastic. And we see opportunities potentially for those of you who perhaps don't have an active seed collecting program yet. And we wanted to pose another question for all of you. This one was an open-ended response, so we're going to break the rule and use the chat box for this. If you could let us know what native tree species growing in natural areas within a 100-mile perimeter of your garden are currently threatened with a significant loss of genetic diversity by an invading insect or disease. If you could use the chat box to type your replies, we'll be paying attention to those and they may have some projects that we could collaborate on in the coming months and years. So while everybody's responding to that, I wanted to pose a question to Shauna. What specifically makes seed banking, in your opinion, such an efficient tool for tree pathogen mitigation?
Hey, thanks for the question, Pam. Yeah, so we can conserve a high percentage of genetic diversity of a species, what some people describe as maintaining a genetic safety net, threatened by a pathogen before it's lost, including those ones that may have resistance, in an extremely efficient and cost-effective way, since seeds are tiny. So like, think about the difference in resources it would take to maintain nearly 100% diversity of a species through seed banking versus maintaining large trees, for example, across a botanic garden or multiple botanic gardens in an orchard type of setting. And then in addition, thinking about trying to save that genetic, maintain that genetic safety net of a species while there's a pathogen, then those trees in a garden may also be under threat by that same pathogen. So it's exponentially more cost effective to house a collection in a seed bank with respect to time, dollars, overall when you think about labor costs, water, electricity, space. And this is possible with seeds that are orthodox for long-term storage.
Oh, fantastic. Well, thanks so much, Shauna. That makes great sense. And I have a question for Tim now. What do gardens need to do to participate? If they wanted to, if they're hearing all this and they want to jump in and participate in biodiversity conservation, what would you suggest to them?
There's a lot of resources that are nice to have, but I think the poll question today is very well chosen on seed banking. If you've got a seed bank, it's the simplest, easiest, low bar way to get involved in conservation. And if you're one of that 37% of gardens that don't have a seed bank according to the poll, please allow me to talk to your director for you. Committed staff, of course. There's a lot of resources needed, and having the committed staff will help find those resources themselves. I have a budget of 0 for field work, yet I get it done. And finally, one of the things I appreciate about conservation is how collaborative it is. So think about your strengths and what you can throw into the stone soup pot. Another piece of low-growing fruit is on these APGA grants, we're looking for distribution sites, as I mentioned in my talk. Become a distribution site, grow some plants.
Fantastic. Oh, thanks so much. Thanks for that answer, Tim, and thank you all presenters, Kevin, Shauna, Richard, Tim, for participating in this session. If people have more information that they want to know, please contact the presenters directly, and we can continue this conversation. If you want to get involved, please do so. We would love to talk to you. So we wanted to let you know this session is eligible for continuing credit hour from the International Society of Arboriculture. The ISA course code is PN20161. We're also putting it in the chat box. For anyone with CEU credits, please contact Tommy Rosenbluth at our association at trosenbluth@publicgardens.org. And you can access the presenters' slides as has been chatted. We've got those in the virtual conference community resources. So please stay tuned for a few quick announcements from the association, and there will be plenty of time for you to get to your next breakout session at 20 after the hour. So thanks everyone for coming. Take care, have a good day.
Hello, everyone. I'm Richard Duran, your association's Marketing and Communications Manager. Over a decade ago, the association launched an awareness campaign with Rainbird called National Public Gardens Day, celebrated the Friday before Mother's Day to focus attention on the important role of public gardens and conservation. Last year, the association refreshed this initiative to create National Public Gardens Week, expanding it to a 10-day celebration starting Mother's Day weekend. A record-setting 200 gardens participated. This year, the global pandemic forced us to change the way we celebrated National Public Gardens Week. With over 90% of our member gardens closed in May, we focused more on supporting gardens' recovery over the spring months and promoting their virtual resources like online classes and tours. Thanks to you, we reached a new high of 230 participating gardens this year, but we're not through. We continue to drive support through your gardens through our social media feeds in what we are calling National Public Gardens 2020. You can find more information at npgw.info. We hope you will enjoy this short video celebrating this year's week, and we look forward to returning to National Public Gardens Week next year, starting on May 7th.
The usual practice during National Public Gardens Week is for us to open our gardens free to the public. But this year is a little different. It is not business as usual. In fact, it's business unusual.
Aloha from National Tropical Botanical Garden Headquarters on Kauai.
Hello, I'm Joan Thomas, your association's Director of External Relations. I really miss seeing all of you this year, and I look forward to getting together next year with you. The spring has been hard on all of us, but I've also heard many stories of resilience, how you kept programs going, used technology to stay connected, and kept members, your coworkers, and your community connected to you. It's also been a time when we've seen funding come to the aid of gardens, to individuals, and to community organizations. For this year's campaign, we wanted to pause and focus on what makes you stronger as a person, as a member of the association, and as an institution. We know the next few years are ones of recovery, recalibration, and reassessment, and hopefully rejuvenation for you. There are over 9,700 of you out there, members of this association, and we want to make sure that over these next few years, you have all the support you need to not only recover, but to thrive. Our goal in establishing the 9,700 Strong campaign is to support each and every one of our 9,700 members over the next few years, to help us stay responsive and to help you stay resilient and constant in your membership with us. We started 9,700 Strong in mid-April and got early support from conference sponsors and with the help of additional champions and our exhibitors. PlantSnap, one of our affinity partners, joined in the campaign and sweetened the deal by offering a free unlimited term pro download of the PlantSnap app to donors giving $20 or more. So for the past 2 months, hundreds of people all over the US have been donating to support you, our public gardens. We're now extending the PlantSnap incentive to all of you as we close in on our campaign goal of $100,000. We're so close. I've been so amazed at the outpouring of personal support to help your fellow members, whether scholarships or member resilience. And so I hope that during this conference, you'll make a gift and join with me and others in reaching this goal. Follow the links on your conference dashboard to give, or you can text 9,700 Strong to 41444. We'll be showing this campaign thermometer throughout the conference to show progress to and hopefully beyond our goal. Thank you so much for being with us and enjoy the conference.
Hi, I'm Jamie Riccio, the association's Member Development Manager. I joined the association in January of this year, and I was looking forward to meeting all of you in Portland at my very first association conference. I wanted to give a special shout out to our sponsors and exhibitors, many of whom have been longtime supporters of our conferences and have stayed with us this year for the virtual conference. And while we don't have an exhibit hall this year for you to walk through, I did want to remind you to visit our Partner Spotlight section of our conference website. Please take a moment to interact with these members and to view their videos. Some of them have offers and other news to share and are here for you. Thanks and enjoy the conference.